Hello everyone, my name is Natascha Wosnick, and first of all, I would like to thank you all for being here, and also the Elasmo Week organizing committee for putting this together. So today we're going to be talking about the sublethal effects of commercial capture on elasmobranchs and what are the implications for fisheries management and also conservation. So in a perfect world, all elasmobranchs would live in a super balanced environment with healthy populations and a lot of protected areas and places without anthropic pressure and all that. And also, research will probably be more focused on non-lethal technologies, telemetry, and all the things that we love to see on TV shows. Um, However, unfortunately, guys, this is not the reality of underdeveloped and developing countries such as Brazil. So I added some photos of commercial fisheries here in Brazil, actually landing points of commercial fisheries here in southern Brazil. And you guys can see that there's a lot of animals being caught daily, several species, different life stages. So it's kind of difficult to deal with uh, conservation, elasmobranch conservation here because our problems are huge. So our situation is this one. We need to deal with intensive fishing pressure coupled with lack of monitoring, flaws in legislation, also some social and cultural aspects related to artisanal fisheries. For example, fishing for sharks is a family tradition that is passed through generations, so it's very difficult to explain to artisanal fisheries the importance of conserving sharks and rays. Also, fishing for subsistence is a reality in Brazil, so if a fisher that is poor caught a big shark, it means that his family or her family, we don't have a lot of female fishers here, unfortunately, but it means that his family will have better conditions for a while. And also local demand for shark meat. We have a huge problem in Brazil of shark meat consumption, and some researchers are discussing and publishing data indicating that some international fishing companies are sending this shark meat to Brazil and actually incentiving, giving incentives to shark meat consumption here in Brazil in order to have a way to dispose the meat um, as the main interest is the things that are being sent to the Asian market. And I need to remember you guys that shark meat, it's like a general term, but it also includes ray meat that is sold along with shark meat as cação. That is the commercial name of elasmobranch meat here in Brazil. So all those aspects taken together result in very expressive population declines. And it's very, very difficult to work with conservation in countries like Brazil that have all these situations going on, since as much as we want to work with live animals and do all those super cool studies and scientific capture and all those stuff that you normally see on TV shows, our reality doesn't allow us to do this because we do need to direct our efforts and money to resolve or at least help to um, advance in these specific issues that are very relevant for conservation in a global scenario. The Atlantic nurse shark is a very good example of what I'm talking about. Um, I don't know how many of you live in the United States, but I'm pretty sure that the ones who live there knows that nurse sharks are like a plague over there. There's tons of nurse sharks, populations are super healthy, animals even in poor conditions, environmental conditions are doing great, they're reproducing, and they're even being caught recreationally. So For those who live in North Atlantic, it's very difficult to understand why the species was listed in a higher category in the last risk assessment made by UCN Red List. So the species was listed as non-threatened. I don't recall exactly what was the specific status before, but it's now listed as vulnerable. And this is because situation of South Atlantic populations is very critical. Um, I was able to work in a place for 12 months where nurse sharks occur. Unfortunately, we don't have the species here in Paraná. The lower limit for the species was São Paulo, but they are locally extinct there right now, as they are in Rio de Janeiro, that is the adjacent state going north. And in Maranhão state, we have some population reminiscence, but the fishing pressure and the environmental conditions for these animals are like absurd. You guys have no idea how endangered nurse sharks are here in Brazil. In fact, they are protected by law here, and there's evidence of severe population fragmentation. And when you have a species of global or like cosmopolitan distribution, which is the case of the Atlantic nurse shark, it's kind of difficult to get all those different aspects of conservation in different places and put them together in a global assessment. So although the species is well protected in the North Atlantic, the situation is critical here in the South Atlantic. In fact, for some of you, I believe that this is the image that you have when you think about nurse sharks, right? So clear waters, a lot of animals, all of them very healthy, positively interacting with humans, such a like dream scenario, right? So this is the situation of most North Atlantic populations, specifically on the Caribbean islands. But this is not the reality here in Brazil, for example, and I dare to say other South American countries that the species occurs. This is a photo that I took in 2018 in Maranhão State. As I told you, it's northeastern Brazil, I think. I'm sorry if I'm getting this wrong. So the species is targeted or over there, although it is protected by law since 2014. And this is the reality of artisanal fisheries over there. The animals are daily caught by long lines, and there's no safe place for them. They caught males, females, pregnant females also, juveniles and adults. So it's a species that is well protected in some places, such as Bahamas, Florida, and other places, but is suffering from human activities here in South Atlantic. I just want you guys to understand the differences, the huge gap that we have between some places in the world and the situation that we live in underdeveloped or developing countries, such as Brazil. And although the situation of Atlantic nurse sharks in the North Atlantic is good, it's like a beacon of hope, as it is for other elasmobranch species that are under federal protection over there, and also in Australia, for example. I know, yeah, Australia, we still have some problems, but still, in developed countries, the global situation of elasmobranchs, it's not good. So we had a recent update on the UCN Red List, and now we have 36% of all currently described chondrichthyes in some risk category, meaning vulnerable, endangered, or critically endangered. And guys, the situation is critical for Rhinopristiformis, that is the order of which sawfishes and guitarfishes belong. So there is a paper that was published by Moore in 2017 that I really liked, that talks about warned us about the critical situation of guitarfishes. In fact, the paper was titled "Are the guitarfishes the next sawfish?" Because we now know that sawfishes are in a very poor condition, and the situation is very critical for them. And guitarfishes are going to the same way, are going to the same path. And that is because both species negatively interact with coastal fisheries. They're constantly caught as bycatch, and evolutionarily speaking, they have some aspects that make them more vulnerable to interactions with human activities. So with that as a context, me and my research group have been devoting like a lot of years of research to understand some crucial aspects of guitarfish commercial fisheries in order to direct future management plans for them here in Brazil. And we're going to talk about some of the results that we found in the last years concerning the sublethal effects of commercial capture have on them. So under this scenario, I'm going to talk about the specific situation in southern Brazil, that is the place that I live, more specifically Paraná coast, and how is the commercial capture of guitarfish over here. So here we have three species of guitarfish, two pseudobatos, so the Pseudobatos horkelli, that is the Brazilian guitarfish, the Pseudobatos persellens, that is the cholla guitarfish, and also the short-nosed guitarfish known as Zapteryx brevirostris, okay? So all three species are currently constantly caught by artisanal fleets. They are normally caught as bycatch in fisheries targeting flatfish during austral winter, that is right now, so from June, actually May until September. So it's like a period that is very of a high concern for us. And some of the animals are landed still alive. Mostly short-nosed guitarfishes. For the both Pseudobatos, the situation is a bit critical because they normally don't stand, they are not able to cope with all the stress imposed by commercial capture. So the situation here is that we have a lot of artisanal fisheries that rely on elasmobranchs fishing or other types of fishing, but caught some elasmobranchs during some specific times of the year. And as the animals are landed normally in poorly situations concerning their chances of survival, the fishers normally sell these animals and fetching very low prices, more specifically for the short-nosed guitarfish. When we are talking about Pseudobatos persellens and also the horkelli one, prices are higher, but still not something that justify the trading of these amazing animals, okay? So under the scenario of fishers constantly caught these animals during the winter and trading them for very low prices, considering all their importance to the marine environment, we are studying some crucial aspects for their management, and I'm going to talk about those results a bit further. So for two of three species that I told you guys, we have some specific legislation that prohibits their capture, landing, transportation, and also trade across the Brazilian territory, which is the Ordinance number 445 that was published in 2014 by the Brazilian Ministry of Environment. So this fishing ban protects not only Zapteryx brevirostris, the short-nosed guitarfish, but also the Pseudobatos horkelli that is currently listed at critically endangered both by UCN and also the ICMBio, that is the Brazilian government agency responsible for manage biodiversity. Regarding the Pseudobatos persellens, although the species was placed in a higher risk category last year, December last year, I think, it is still not threatened in Brazil, so its capture is still permitted, as so it's straight. So under the scenario of pro, of prohibition of capture and all the stuff regarding commercial capture, there's still a lot of animals being caught and being landed, especially as I told you guys during the winter here. So this is a photo from 2 weeks ago. We were on field, and those are individuals that were caught by one artisanal fishing vessel in one fishing campaign. It was 78 animals, so those are the females here close to me, and the smaller portion in the right photo are the males. So we have some specific fluctuations during this period of capture, intense capture. So normally during May and June, males are predominant, and from July to September, pregnant females are predominant in the catch volume. Okay, so this on the left, it's a photo of the individuals that we were able to recover with the fishers and promptly release. So some of you might thinking that this is very unprofessional, have them all like stocked like this, stocked pile up like this, and they're all alive, okay? And then why you guys are like leaving them outside of the water for so long? So let me tell you guys, the short-nosed guitarfish is extremely robust and very resilient. They can live outside of the water for up to 6 hours. And when we are transporting like 78 individuals at once, it's pretty much impossible to have good water conditions to ensure that they are not going to be intoxicated by their own toxic products. They are like releasing, I'm sorry, during stressful situations such as toxic ammonium and also another nitrogenic compounds. I'm sorry, I don't know if I'm saying this right. So, for example, for this species, we know that we need at least 100 liters per animal to have like a good water quality. Okay, so it's pretty much impossible to do this with the amount of money that we have right now to invest in this type of activity when we normally buy all these animals with our own money. We have some fishers that are our partners, so they donate a lot of animals, but right now we are trying to expand our network and trying to invest in other relationships in other places, other fishers, in order to create like a wider network to get these animals and release them back to the nature. Okay, so this might not seem as professional as we wish, but still it works. Our survival rates are up to 96%, even with pregnant females and also sexually active males right now, as we are improving our protocols of getting the animals from the fishers as soon as possible and in places that are closer to the coast than the first landing point that we used to work. Okay, so the results that I'm going to show you on the sublethal effects of capture in a minute are regarding the landing points that we are not working as much as we used to right now. Okay, so those photos, as I told you guys, was from 2 weeks ago, so we were able to buy and rescue and release in 3 days. It was 185 guitarfishes. So from all the species that I told you guys, both pseudobatos and also zapterics, with the main portion being of zapterics. Okay, so what we do, and it's working very decently, I can say that. I mean, it's not the ideal condition. In a perfect world, these animals will not be caught at all because they're protected by law, but we need to understand that fishing communities have their own culture and all and their own like social demands and economic demands, and we need to respect them and try to include them in our research and conservation actions. I really believe in that. I really think that we cannot do conservation in developing countries without the support and participation of fishing communities, okay? So what we are trying to do is release as much as animals as we can. And since 2010, since 2010, we were able to release about 120, I'm sorry, this number of guitarfish back to the wild, okay? And also, as I told you guys, specifically for the short-nosed guitarfish, post-release mortality are very low. So we are having a lot of success in releasing these animals. Unfortunately, we don't have enough money to tag them, both with like macaroni tags or acoustic tags. So now we need to go further and try to understand what is going on post-release. If these release activities are being as efficient as we think. As I told you guys, we think so because we are now releasing them in a place that we can monitor for at least a week. So normally when they die from the stress of capture, transportation, and release back to the nature, they come back with the tides. So we are able to recover them and get all the information, do necropsies, photograph them, also take some blood samples to try to understand what is going on and why they are dying. And we now have a very robust scenario, and we have robust data, and we are now understanding exactly what is going on with this animal with these animals physiologically speaking, okay? However, as release seem like super cool and also a conservation measure that is very promising. However, there are some sublethal effects that are taking place, such as abortion, that is, in my opinion, the most graphical one. The first time that I saw a female aborting her embryos was very traumatic for me. And I have a photo here that it represents very well the situation. So here we have a female that was alive like minutes before we took the photos. Unfortunately, she died after after expelling her last embryo. So we can see here one embryo, a dead embryo by her side, and another one being expelled at the moment of the photographic record. And we also have here another photo of another embryos from another female that were reported by our research, that were recorded, I'm sorry, and then reported by our research team. So abortion is a problem not only because causes the loss of a litter, so impairs recruitment, but also because according to our results, it is followed by high rates of maternal mortality, as I'm going to show you guys in the next slide. So during my PhD, I was able to monitor and investigate the abortion process in more details. And apart from using some physiological markers to understand what was going on with the females, I don't know if I told you guys, but I do work with physiology. This is my main research line, and I like to apply physiological tools to inform elasmobranch conservation. So what I did during my PhD was to buy as many as pregnant female as I could from artisanal fishers. And I want to highlight here that all the studies that I'm going to show you guys from now on are a result of a partnership with artisanal fishers and also reflects the reality of commercial fisheries on on our study region, okay? So what we saw on this study by monitoring the pregnant females for 48 hours is that the first hours after capture are the most critical ones, as the main portion of abortion occurs at the first hours after capture. and there's a tendency of reducing of reducing the amount of abortions after a while. The problem is, while the abortion is a abrupt response to capture stress, meaning that occurs right after the animal is exposed to this very stressful situation, the mortality of the mother, the maternal mortality, occurs several hours after the abortion event. So even if you see a pregnant female aborting their embryos and release this female alive, and we have some reports of fishers saying that when they say that female is aborting, giving birth, they say giving birth, they help her, so they do some physical pressure on their bellies to expel the embryos faster, and they release the female, and they think this is a good action. However, the abortion is occurring, they are applying additional pressure to the abdominal cavity, and there's also this very sad result that we saw. That is the mothers, they die, most of them die after consecutive abortions. Actually, it was 60% of them. Please note that the results are 75% here in the graphic, but that is because around 50% of maternal mortality was due to transport to the laboratory. Unfortunately, it was like a human error, but now we understand that the rates are lower than that, but still very high, considering that more than half of females that abort their embryos die soon after. And regarding the physiological markers, I'm going to spend a little bit more time on this graphic here, because I do want to explain you guys the markers that we are using right now. Those are all stress markers. Some of them are very known and widely used across the world, especially lactate and phosphorus, actually potassium, I'm sorry. So those are markers that are very reliable, as both always increase with capture stress, okay? So every time that an animal is super stressed, lactate levels will be super high, as so potassium, I'm sorry. I don't know how to say that in English. And what we tried to do here, what we did here was to validate some markers for pregnant females for the species that we are talking about. We need to remember that stress markers are very species specific, so we always need to analyze and be careful when we draw conclusions about stress, mortality, or whatever when we are talking about physiological markers, okay? So in the case of pregnant females of Zapteryx brevirostris, pH seemed to be proved to be a very reliable marker, as we found decrease on plasma pH upon lactate increase in plasma, okay? So it was negative correlation between both, that it was expected, as lactate is a marker that increases when you have a situation of blood acidosis, which means that the pH will drop, okay? So it's very interesting to see how those markers relate under stressful situations for the species, and more specifically for pregnant females of the species. And also we decided to test urea, that it was very reliable for females, as I don't know if you guys know, but elasmobranchs have a very specific osmoregulation profile that is super different from bony fish and also mammals, or pretty much any aquatic vertebrate, okay? Because they rely on urea, that is a compound that it can be toxic in high concentrations. But elasmobranchs evolved in a way that they can store huge amounts of urea without being lethal for them, okay? And urea, it's very important for osmoregulation. And when we talk about stress, we are talking about the loss of the homeostatic balance. And this is directly linked with osmoregulation processes, okay? So, as urea is the main component of osmoregulation in elasmobranchs, it's really important to measure urea concentrations in plasma in order to see if this balance is being lost and not just some transient alterations that are not as serious as the complete loss of homeostatic balance, okay? And also we tested for phosphorus, and it was very interesting to see how promising and how reliable phosphorus is, okay? As potassium, phosphorus is a intracellular marker, and when they increase in plasma or blood, it means that the animal is presenting cell rupture. That is very concerning as it is a lethal situation. Okay, so once again, it's very important to analyze very carefully the stress marker that we are using concerning physiology, as they can change a lot, and a stress marker to be reliable, it needs to be constant, which means it needs to always have the same response, okay? For example, lactate and potassium and also phosphorus will always increase if the animal is exposed to a stressful situation. pH under very severe stress will drop in values that are very significant. Sometimes it's kind of difficult to work with pH because a zero point 0.1 drop could be enough to cause a series of damage, of a cellular damage, but it's not enough to be detected by statistics, okay? So that's why we need to discuss the data very carefully. And what we saw for the pregnant females. So first of all, we have a comparative approach for this study. So the first group that you guys are seeing in all the graphics, the pink one, light pink, it refers to non-pregnant females, so females that were caught by artisanal fishers outside the reproductive period and were monitored by our team for the same amount of time that the pregnant females that suffered consecutive abortions, okay? The second group, represented by the green color, represents the pregnant females that survived after consecutive abortions, and the third group refers to pregnant females that die after consecutive abortions, okay? So what we saw for these 5 markers that we decided to use is that the females that survived to the stress of capture and subsequent abortion were closer to the females that were not pregnant, meaning that physiologically they were able to recover fastly than the females that die. So the females that die, that are represented by the gray box plots here, were not able to recover from the stress suffered by capture. So they had a allostatic overload that we call allostatic overload, meaning that they lost completely the homeostatic balance and weren't able to recover in time, which lead them to death. Okay, so it's very interesting to see how, in a comparative way, we can see exactly what is going on with females upon consecutive abortions, and why some females are able to recover while others are not. So as take-home messages, pregnant females of short-nosed guitarfish, even if released alive, suffer the sublethal effects of capture, and specific protocols are needed during this period of great vulnerability. So the next question was, if the female survives, can these effects, the sublethal effects, aka the abortions, can impair fertility in the mid and the long term? And to answer these questions, we focused on uterine alterations on the macroscopic level, okay? And we did this through the use of ultrasound by comparing pregnant females with females that aborted their embryos and were aborted their embryos but survived, okay? So this study is also published. I can send you guys if you want to. And I'm not gonna enter into very specific details on methodology and stuff. But what you need to see is that we were able to characterize some macromorphological conditions of the uterus of pregnant females. So here we have the right uterus and the left one. Elasmobranchs always have two. They are not always functioning at the same time, but for the species it is, okay? And so we have here some embryos that are detected to the midline, and also the egg capsule, as this species is viviparous present lecitotrophic viviparity, okay? So there's no placenta, and the animals are kept, the embryos are kept in egg capsules and are nourished by the yolk sac for the whole embryonic development. So we can see the egg capsule here, the midline of the embryo, and also we can see some characteristics such as a dilated uterus, a uterine wall that is very thick, yeah, I think that's correct. Thin is, okay, so thick, and also a good amount of uterine liquid over here, okay? So this is a normal situation for a pregnant female of short-nosed guitarfish. And what is the scenario, what is the condition of a uterus for a female that aborted her embryos and survived to the stress of abortion? This is what it looks like. So we still have a very dilated uterus with a lot of uterine liquid and also a very thick uterine wall. So the female maintains the characteristics, the macromorphological characteristics of the uterus, even after consecutive abortions and a long time. Not a long time, it's like short term, but still. So what we can see here is that the situation is kind of critical because we don't know for sure if these females are able to recover their non-pregnant characteristics, okay? So as we all know, vertebrate uterus passes through a lot of changes to be able to receive an embryo and give all the good conditions for this embryo to develop, okay? And when we have something that alters for good these characteristics, the morphological characteristics involved with fertility and ability to keep an embryo under good conditions for development, this is a problem because even if you release this female and it survives to the stress of abortion, the female might not be able to reproduce once again. So take-home messages: after abortion, females exhibit significant uterine alterations compatible with physical trauma. And, for example, in mammals, such alterations are associated with reduced fertility in the long term. So we need to keep an eye on it and do further research to understand if this is a situation, a long-term situation, so fertility it's not as good as it used to be, or if the female is able to recover for the next reproductive period, for example. So now we understand that for the short-nosed guitarfish, females are very sensitive to capture. They abort their embryos and they normally die after. And also the females that survive normally present some macromorphological alterations that can compromise their fertility. But what about males? So with that as a context, my back then graduation student Aline Prado developed a study considering the males for the same species. So in order to analyze the period of greatest vulnerability for males, Aline decided to investigate some morphophysiological aspects of male, specifically during the copulation period. That is represented by the phase inside the reproductive period in which males present semen in their seminal vesicles, okay? And also it's in theory the phase in which males will expend more energy to produce sperm and also to copulate with females, perform some specific behaviors that are related to this phase itself. So in order to do this, Aline decided to analyze not only the markers that I did for the pregnant females back then in my PhD, but also to add another markers to see if they were good enough, not good enough, but if they were reliable for males, okay? That are sodium, chloride, and also plasma osmolarity. And what she saw is that males are as vulnerable as females in terms of physiological response, and they behave very similarly with females during the pregnancy. Okay, so here we have all the markers that she analyzed in the plasma of the animals, and the comparative approach was also used in this investigation. So the first box plot of all graphs refers to the males outside the reproductive period. The second one refers to the males that survived to the capture stress during the copulation phase. And the third one refers to the males that die upon capture stress during the copulation phase. And you guys can see that in general, overall, all the males that die of capture stress during the copulation phase presented alterations that were significantly different from live males and also males outside the reproductive period, indicating that these males lost completely their homeostatic balance. These males, right? I say males, okay. And also weren't able to recover from levels that are compatible with life, okay? So also it was very important to analyze males as our survival rates were even lower for males comparing to females. It was around 68%, the mortality rates for males, while the mortality rates were 60%, okay? Excluding the 15% upon transportation that we had for the pregnant females. So it was 68% for males, the mortality rates, and 60% for females, indicating that the reproductive period, it is in fact the period of greatest vulnerability for both sexes. I don't know if I told you guys, I think I did on the release slide, but our survival rates outside the reproductive period are very high, are around 100%. It depends on the fishing gear employed and also the time trapped in the fishing net and also the time to transport these animals from the capture site to the landing site, but it is always around 100%. Now we are getting higher survival rates even for pregnant females and sexually active males, but it's because, as I told you guys, we are working with different fishers right now that delivers animals in better conditions. So we are now need to understand what are the specific difference in handling protocols that are allowing these animals to survive more even during the reproductive period. So it's very actually promising, because we thought at once during these studies that I'm showing you guys that the survival rates were always lower, but it's not the case, and now we need to discover why it's not. So Aline was also able to analyze some energetic markers that I wasn't able to analyze during my PhD, during logistical difficulties. So she chose a physiological marker, aka triglycerides in plasma, and also a morphological proxy, which is the Fulton condition factor that is widely used for fishes in general. And what we saw was that both live and dead males during the copulation phase, they mobilize more energy from hepatic stores, so liver stores, in order to cope with the stress of capture, while males outside the reproductive period don't need to mobilize as much as energy from stocks to be able to cope with stress. And although there's no significant difference between live and dead females inside the males outside the whoa, let's do this again. Although there is no significant difference between live and dead males inside the copulation phase, you can see a tendency of higher levels of triglycerides, circulating triglycerides for dead males, indicated, indicating that these males need to mobilize more energy, but still it is not enough to prevent them to die. And for the Fulton condition factor, our results were expected actually, because as I told you guys, in theory, the copulation phase, it's a period that the males need to like work a lot. So it is expected that they will be thinner, which was the case, okay, for both live and dead males, with no significant difference between both, but a significant difference when compared to the males outside the reproductive period. She also saw some physical alterations that are compatible with stress. And I am highlighting the flaring condition, which is the opening of the clasper, of the extremity of the clasper as a result of intense stress. And males normally do this as a way to defend themselves. And this condition is normally associated with high levels of lactate and also phosphorus, which indicates a lethal condition. So flaring is a good proxy to be used in future assessments on onboard monitoring, for example. If the male presents this condition, it is probably too late to save him, unfortunately. And also we decided to compare the results from males and females from my study with Aline's study. And we realized that physiologically, considering only these 4 markers, as I didn't analyze the rest of them back then, as Aline did later. So guys, just quickly explaining. So we have 4 graphs here, and we have each graph separated in three blocks, okay? So the first blocks refers to males and females outside the reproductive period. The second one, those that survive it after the stress of capture, and the last one, those that die after the stress of capture and abortion, okay? So what we saw is that females and males respond very similarly to the stress, but some expressive difference was observed regarding the specific markers. So for example, lactate increases more in females that die than in females than in males, and urea is the opposite, while drops for females increases for males that die, okay? Phosphorus the same, with sex-dependent patterns. And this is very important when we consider that management plans does not always or never actually consider sex as a variant that needs to be taken into account. So take-home messages for this specific investigation. We now know that males are more vulnerable than females during the reproductive period, more specifically during the copulation phase. We also know that energy mobilization is not enough to cope with capture stress, at least for males, and specific protocols are also necessary for them, meaning that a management plan will benefit both females and males of short-nosed guitarfish if measurements are adapted to the reproductive period. So to add another piece to the short-nosed guitarfish puzzle, Aline, during her master's, decided to investigate what was going on with the embryos of short-nosed guitarfish. And she did that based on two main theories that are very common for pregnant mammals regarding the fate of both mother and litter, considering a situation, a very stressful situation, okay? So the first theory is self-sacrifice, which means that the female will sacrifice herself in order to increase the chances of their pups, of her pups or litter or embryos, neonates to survive. And in an opposite trend, there is the theory of self-preservation, in which female will try to save herself at the cost of losing her litter or leaving her cubs, pups, whatever behind. So those results are not published yet, but I'm happy to discuss some aspects with you guys, and I can also send you Aline's email if you want to talk to her to clarify some aspects that I'm going to present now. So Aline was able to detect capture-induced parturition in several stages of development for the embryos, and she was also able to identify a whole new category that is called retention. So very quickly, when we are talking about parturition induced by capture, we have two main categories. The abortion one, that is the main category for the short-nosed guitarfish, which is defined by the expulsion, the premature expulsion of litter, but all the embryos are already dead. While the premature birth, as we all can figure it out regarding the name, is the premature exposure of embryos that are still alive. But it depends, like if they're going to survive or not. It depends on the stage of development they are being that they are being released. Okay, expelled actually. And retention is defined by the inability of a female to expel her litter, and sometimes they can stay alive inside the uterus while the female dies, or it can be the opposite. The embryos will die inside the uterus, and the female will survive. And this can have a lot of like problematic complications such as infections and subsequent female mortality. Okay, so according to Aline results, abortion can occur in all stages of development, while premature birth is restricted to the early stages of embryonic development. And retention, although occurs in various stages, it's more common for embryos in late stages of embryonic development. So we think that retention is more related to the inability, physical inability to expel the the leader without proper physiological activation of some specific pathways, such as hormones that will help the female to contract the uterus to release these animals. If it's something that occurs very quickly, she's not able to do all those uterine contractions necessary to expel a bigger embryo, okay? And regarding the physiological markers, I'm going to show you three, I think. So urea is the first one, considering the importance of this molecule for elasmobranch homeostatic balance. And here we have three graphics, one for abortion condition, another one for premature birth condition, and the last one for retention condition, okay? So we have one box for mothers, one for uterine fluid, and one for embryos for all three situations. And what we saw is that the alterations observed in the plasma of the mothers, it's very similar to the ones observed in the embryos, in the plasma of the embryos. And this is a strong indication that they are experiencing the same magnitude of stress. As for lactate, the situation is very similar for both mothers and their litter, but we can see that the concentrations are lower in the uterine fluid compared to both groups, meaning that probably the lactate concentrations observed in the plasma of the embryos are of their own. So there's no effect, at least directly, of the lactate, the circulating lactate on the mothers, on of the mothers on the embryos. Because if lactate from the mothers were passing to the embryos, the concentrations of the uterine liquid will be higher. We also analyzed glucose for both mothers and embryos, and also in the uterine fluid, in order to understand. We normally don't use glucose as a marker, as glucose varies a lot, not only between species, but also between conditions. For example, there are some animals that respond positively to glucose levels regarding capture stress, so they will present higher levels upon capture stress, but there are some situations, species, animals that will uh, present lower levels, like there will be a decrease in glucose levels. So as I told you guys, a stress marker to be reliable, it needs to behave always the same way. So that's why we don't think glucose is a good stress marker, but still can be used as an energy mobilization marker, more specifically to see how the energetic dynamics are interacting with the stress imposed by capture, for example. What we sought for glucose is that there's no difference between the response for both mothers and embryos. And actually, we normally, when we talk about capture stress of capture concerning energy mobilization, according to their diet that is mostly based on protein, elasmobranchs will firstly respond with higher circulating levels of ketone bodies. And then the second line of response to the stress will probably be triglycerides, considering their diets that are very rich in lipids. But also there's a slight, like not slight, but like a smaller component of glucose on this response, as part of their diet is composed by carbs, but it's not as significant as it is for mammals, for example. So it's kind of difficult to deal with glucose and understand what is going on and also discuss these results. But here we can see that glucose response for both mothers and embryos was the same, but it was significantly lower in the uterine fluid for both for all the situations. As for triglycerides, the situation is a bit different, and we were able to see that embryos present higher concentrations upon capture stress than their mothers, and also than the uterine fluid. So in the first graph here, the pink one, there's no statistic difference as the green one. But the third one, we were able to detect a statistical difference between mothers and their embryos. But for the first graphic here, the abortion one, you can see some outliers around 500 milligrams per deciliter of triglycerides on the embryos, and also some outliers for the females, meaning that although no statistical difference was observed, it is possible that there is a difference between the groups that is not seen on statistic tests, okay? So what those results mean? It means that the triglycerides are the main fuel for embryos to cope with stress, which is not the case for the mothers. Or it could be the case for the mothers, but it's not as strong as the response it is for the embryos, okay? And why is that? We think it's because embryos only rely on their yolk, so there's has to be a main component on short-nosed guitarfish yolk that make triglycerides the main fuel for embryos in the development stage that rely solely on the yolk reserves. Aline was also able to analyze dissolved oxygen in the uterine fluid for both retention and abortion categories and compare those results with seawater as a control group. She did that because there's a theory from Voren and Rincon that embryos die from hypoxia, meaning that lower concentrations of oxygen in the uterine fluid will make it difficult to embryos to breathe and do all the gas exchange that is necessary to maintain their vital functions. And in fact, the results that we got corroborates, like confirm this theory, as for both retention and abortion categories, dissolved oxygen was very low compared to what was expected for the uterine fluid, considering that it needs to be similar to seawater. And what are the take-home messages for this investigation? So embryos seems to experience the same stress as mother, or at least it occurs in the same magnitude for both. And also embryos seem to contribute to capture-induced parturition. More specifically, guys, remember that I show you that lactate, embryonic lactate is very high. So in this specific condition, when you have a lot of circulating lactate inside the body fluids of an animal, it could lead to a metabolic acidosis, that is a drop in pH. And drop in pH normally alter all of the metabolic dynamics of these animals, leading to a higher waste of toxic compounds, which can result in a toxic environment, uterine environment, that could also lead to a higher rate of abortion, or at least consecutive abortions. So it's not a situation that is controlled or it's caused only by the females. What is happening with the embryos is also contributing to the situation as a whole, okay? And also, self-sacrifice and self-preservation theories do not seem to apply to the species. That is because if self-sacrifice was the case, mortality rates for the embryos will be lower, or at least a part of the embryos that are being expelled will survive, at least for like a couple of hours, which is not the case. Embryo mortality was 100%, so there's no meaning to have a self-sacrifice if your embryos will not survive. And I'm talking about evolution, not something that the female decides to do. As for the self-preservation theory, it does not apply to, as if you're like killing your leader in order to ensure your survival, mortality rates will be lower than 60%. So it seems that is not the case for the short-nosed guitarfish, at least. So finally, what we learned from all these years studying the sublethal effects of commercial capture on the species is that the impacts of fishing go far beyond mortality, so we need to consider all those other things that are happening with these animals apart from just mortality rates. Attention must be paid to the reproductive period, as even if animals are released alive, as in the case of short-nosed guitarfish, sublethal effects may occur for both males and females. And for the species, pregnant females are less vulnerable to capture mortality than sexually active males, however, possibly at the expense of litter loss. Also, the stress suffered by embryo seems to contribute to the uterine changes that lead to abortion, indicating that the female has little control over this phenomenon. And finally, as the species is caught as bycatching fisheries targeting flatfish, seasonal closures unfortunately are unrealistic. That said, it is necessary that release protocols to be adapted for the reproductive period, as well as alternative ways to reduce capture, such as fishing exclusion devices, but always working in partnership with artisanal communities or fishers in general. Okay, so that's it. Thank you so much for your attention. This is my email and also our Instagram profile where we normally post our research results and also some of our release. Thank you. Bye bye.